So our third lecture is a brief introduction of gene cloning and gene expression. अब बायोकेमिस्ट्री में हमें इसकी कहाँ ज़रूरत पड़ती है कि जब भी हम जीन को मैनिपुलेट कर रहे होते हैं, हम डीएनए को मैनिपुलेट कर रहे होते हैं, वी नीड टू मेक प्रोटीन्स आउट ऑफ अ जीन, तो हमें इस बारे में पता होना चाहिए कि हम उस डीएनए के फ्रेगमेंट को जिसके ऊपर हमने काम करना है, जिसके अंदर से हमने प्रोटीन को बनाना है, हम उसकी क्वांटिटी को किस तरह ज़्यादा कर सकते हैं. So to do that, we go towards the gene cloning, and then to make proteins out of it, we go for gene expression and protein expression, and then protein purification. तो आज का हमारा जो लेक्चर है that is a very brief introduction because the details are there in the molecular and maybe in molecular genetics type of courses. But in biochemistry techniques, we are focusing on what we are going to do actually with these techniques. If we are doing chromatography for protein purification, why are we actually purifying the proteins? And where are these proteins coming from? और उसके बाद if we are let's say doing agarose gel electrophoresis to see the DNA, but why are we actually seeing the DNA? But we need to see the DNA. So इस तरह की कुछ बातें जो कि हमें सब्सिक्वेंट लेक्चर्स में पढ़ेंगे. हम agarose gel electrophoresis पढ़ेंगे, हम SDS पेज पढ़ेंगे और हम इवन क्रोमेटोग्राफी में भी पढ़ेंगे. तो उसको अंडरस्टैंड करने के लिए वी नीड टू नो कि व्हाट आवर गोल इज एंड व्हाट वी आर गोइंग टू डू एंड व्हाट वी एक्चुअली डू while doing these experiments. Okay, so gene cloning is what? Gene cloning is basically a technique to multiply the copies of a specific DNA. For example, आपके पास एक larger fragment of DNA है और आपके पास इसमें से let's say this is your gene A. आपने इसको फोकस करना है कि आपको इसमें से प्रोटीन चाहिए, that is प्रोटीन A. आपने इसको किस तरह से एम्पलीफाई करना है, and then how would you perform the central dogma to get the protein out of this gene. Okay, so what we do is our basic, let me just get a white screen. So what we do is this is our complete overflow of how we clone the genes. So what we have is for example we have the DNA. For example, this is the DNA, the double-stranded DNA, and let's suppose this is our gene. We'll say this is gene X. Okay, so सबसे पहला पार्ट है कि हमने इस जीन को बेसिकली इसको एम्पलीफाई करना है. सो टू एम्पलीफाई जीन वी डू व्हाट वी डू इज वी डू पीसीआर. पीसीआर क्या करता है कि वी मेक प्राइमर्स. फॉर एग्जांपल दिस इज आवर जीन, दिस बॉक्स इज आवर जीन, और ये आपका कंप्लीट डीएनए है. हमने एक फॉरवर्ड प्राइमर एंड रिवर्स प्राइमर, दीज आर द फ्रेगमेंट्स दैट एम्पलीफाई द डीएनए बेसिकली. सो इसके बाद इसकी बहुत ज़्यादा फ्रेगमेंट्स आ जाएंगी. ठीक है, नाउ दिस फ्रेगमेंट of this gene is basically outside the body. ये हमारे पास ऐसी ही जैसे एक केमिकल है बेसिकली. सो हम इससे प्रोटीन नहीं बना सकते जब तक हम इसको एक लिविंग सिस्टम में ना डालें. सो हाउ डू वी मेक प्रोटीन? द सेंट्रल डोगमा इज यू हैव जीन, this undergoes transcription, this makes messenger RNA, and this messenger RNA makes proteins by translation. ठीक है, तो we have to perform all these steps to get the protein out of this gene. तो अब हम क्या करते हैं, हम अभी इस स्टेप पे हैं. we have the gene. What we have to do is we have to go through all these steps all the way to get the protein. ठीक है, so we have gene, our gene X. हमें क्या करना है कि इस जीन को we have to add it inside a bacterial plasmid, और उसके बाद हमने उस bacterial plasmid को डाल देना है in a bacterial cell, and that cell will divide. Okay, so this is our main goal. To do this, we have to follow several steps. What are the steps and what these steps do? We're gonna have a very brief introduction about those steps. So this is our gene. This is our gene, the gene A. हमने क्या करना है कि हमने इसे एक बैक्टीरियल वेक्टर में डालना है. So vectors are basically a circular DNA. So if it's a DNA, it should have some nucleotide bases, and these nucleotide bases are A, T, G, and C. This circular DNA is basically made up of A, T, G, C, 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 G, A, C, A, T, G, C, and so on. हम क्या करते हैं कि we have some sort of molecular scissors. अब scissors क्या करती हैं? Scissors have the ability to cut something. So molecular scissors can cut through a molecule. So what we do is we have these. If I stretch open a plasmid, so a plasmid, for example, this is the plasmid, and this has A, T, C, 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 G, C, G, C, T, A, C. हम क्या करते हैं कि हम हमारे प्लाज्मिड को दो साइड से, फॉर एग्जांपल दिस साइड एंड दिस साइड, यहां से कट कर लेते हैं. दिस इज कॉल्ड रिस्ट्रिक्शन. नाउ these are DNA bases. Okay, these are nucleotide bases. So if this is cut from this side, for example, so if this is cut from this side, we have a hole that can be used to manipulate DNA. So what we do is, for example, this is our DNA, this is our cut DNA. I'm gonna make it more easy for you. So this was the DNA before restriction, and after restriction, this DNA looks like this. Okay, let's make it single stranded. So it looks like this. अब ये जगह हमारे पास है जो कि that is open. What we do is we add the same restriction sites, वही restriction site जो कि यहाँ पे present है. I'm gonna mark them small a and small b. This is small a and small b. We add the same restriction sites in our gene using different techniques such as PCR. और अब हमारे जीन में भी यहाँ पे A है जिसे मैं A नेगेटिव कहूँगा, और इसके अंदर भी B है जो कि B नेगेटिव कहूँगा, and then we can easily place our gene at this spot, which this A negative will react with A chemically, and this B negative will react with B chemically. And what we will get at the end, that would be, I'm just gonna draw it. So A negative, B negative, B. So this is our gene X. So this step is known as the ligation step. Ligation is basically the joining of DNA, the joining of part of DNA, part of the gene, and the restricted vector that has a part of DNA cut by different restriction enzymes. Joining together is called ligation. The step is called ligation. So the next step is, now we have the plasmid that has our DNA attached to it. This is the gene X. Now अब हमने इस प्लाज्मिड को बैक्टीरिया के अंदर डालना है, और इस डालने को हम कहते हैं ट्रांसफॉर्मेशन. सो व्हाट वी डू इज, हम एक बैक्टीरियल सेल लेते हैं, मोस्टली इकोलाई सेल्स, इकोलाई बैक्टीरिया सेल्स. हमने प्लाज्मिड को अंदर डाल दिया जिसमें हमारा जीन लगा हुआ है. दिस वन इज आवर जीन फॉर एग्जांपल. अब क्या होगा कि इस बैक्टीरिया ने, वी नो दैट बैक्टीरिया डिवाइड वेरी फास्टली एज कंपेयर टू नॉर्मल ऑर्गेनिज्म. तो बैक्टीरिया डिवाइड करेगा. दिस इज डिवाइडेड बैक्टीरिया. दिस इज फर्दर डिवाइडेड इनटू more bacteria, and so on. और इन सब बैक्टीरिया में हमारा recombinant plasmid और recombinant vector, recombinant that has gene plus the vector we go. This will grow inside each of the bacteria. So this is how we will get a large amount of our gene. पहले हमारे पास सिर्फ एक जीन था. Apparently हमने इसको जस्ट एक प्लाज्मिड में लगाया, बैक्टीरियल प्लाज्मिड में, एंड देन वी ट्रांसफॉर्म्ड इट इनटू बैक्टीरिया. अब हमारे पास, इफ दिस इज 1, this is 2, this is 3, 4, 5, 6, 7, and so on. So within hours, we will get a large amount of our gene that we can use in further experiments. Okay? So where are the biochemical techniques that are used in this all of these experiments, all this concept. So the biochemical techniques which are used, or which are used are, we have to, let me just, अब हम क्या करते हैं कि once the PCR is done, we have our gene, we go for agarose gel electrophoresis. Agarose gel electrophoresis to see कि हमारा जीन एम्पलीफाई हुआ भी है कि नहीं हुआ. ठीक है. Next step, if our gene is amplified, our gel electrophoresis results are positive, we go to the next step, we restrict our plasmid. So restriction, when we restrict our plasmid, we again go for gel electrophoresis to see if it's restricted or not. Now, for example, if your unrestricted plasmid has a size of, let's say, 3 kb, your restricted plasmid might have the size of maybe 2.89 kb, and it will even change the place in the gel to show you the shape of the plasmid. Now, if a plasmid is in this shape, the circular plasmid, but if you cut the plasmid at two ends, it will change into this shape, which will eventually change into a straight shape. तो इसने और इस फॉर्म ने जेल के ऊपर डिफरेंट जगह पे नजर आना है. अगेन व्हाट वी यूज्ड वाज agarose gel. ठीक है. एंड देन आगे, after ligation, we again go for agarose gel to check if the DNA is ligated or not, because a gene, a plasmid after cut, maybe it's of 2.89 kb. So 2.89 kb plus our gene, if for example, if our gene is of 1 kb, this will eventually become 1 kb, this will eventually become, it will become 3.89 kb. So this and this will have appreciable distance between both of them on the gel. So we can easily say our ligation was actually right, true. Oh, there are other techniques to check that, yes, as well. There are other techniques as well to check those, but we can use gel for this, to validate these results as well. Okay, so let's move on to the slide. So this is about gene cloning, and these are the steps that I have just told you about. Okay.